Hello everyone, welcome to my course on microbial biotechnology. Today we will start module 6, which will deal with industrial and pharmaceutical applications of microorganisms. In the first lecture, we are going to discuss about the microorganisms in industry. So this is briefly the overview of the various things we are going to discuss, divided into 3 sections. In first sections, we will discuss microorganisms in industry, have an overview, and then we will go for the industrial microorganisms. Then we will discuss about microbial cell factories, and then finally the fermentation process, which comprises of upstream processes and downstream processes. So, microorganisms play a vital role in industrial production of a wide variety of goods and services. They are exceptionally valuable due to their ease of mass production, rapid growth, utilization of economic substrates, often waste materials, which can be converted into valuable products, and the vast potential in generating diverse products. Furthermore, their capacity for genetic manipulation has opened boundless opportunities for innovation within fermentation industries. Briefly, industrial microbiology is the application of microorganisms to produce valuable products or perform processes on an industrial scale, including the production of antibiotics, enzymes, biofuels, fermented foods, and waste treatment. This field encompasses a wide range of microorganisms found naturally and also laboratory derived mutants. Initially, traditional fermentation processes relied on a mixture of wild microorganisms present in raw materials or the local environment. Around 120 years ago, efforts began to improve these microorganisms by isolating pure cultures from these processes and selecting the most advantageous strain. In this picture, you can see Louis Pasteur working with wine. Pasteur's contribution to improving industrial microbiology is very significant. Throughout the first 80 years of the 20th century, most fermentation processes primarily used monocultures, which are cultures having only one type of strain or species. These were typically sourced from the natural environment following extensive screening. These microorganisms, regardless of their origin, were subsequently optimized through conventional strain improvement techniques such as mutagenesis or breeding programs to enhance their industrial applications. In the past 2 decades, many fermentation processes have incorporated recombinant microorganisms, leveraging genetic engineering to further improve established industrial strains. One of the main focuses of industrial microbiology is genetic modification of organisms to improve product yield and for reduction of production costs. Industrial microbial biotechnology is a multidisciplinary field within biotechnology that harnesses the inherent biological processes of microorganisms to address various industrial challenges and opportunities. This field is a fusion of microbiology, biotechnology, and industrial processes, focusing on the application of microorganisms to develop and enhance products, processes, and technologies on a large scale. Industrial biotechnology or industrial microbial biotechnology plays a crucial role in producing a wide array of high value products, from life-saving drugs and sustainable chemicals to renewable fuels and clean electricity, all contributing to a more sustainable and bio-based economy. Industrial microbial biotechnology involves using microorganisms to produce a variety of high value products across several sectors. So, if you look into the domain of industrial biotechnology, it basically is multidisciplinary, deriving from the fields of microbiology, biotechnology, and then the industrial processes or manufacturing. The outcome is bio-based chemicals, which gives us synthesis of organic acids, amino acids, alcohols, bioplastics, and specialty chemicals, or it gives pharmaceutical and therapeutics, where we go for production of antibiotics, vaccines, hormones, monoclonal antibodies, and therapeutic enzymes. Then also produces a lot of industrial enzymes like proteases, amylases, cellulases, lipases, and lactases for various industrial application. And then we can use these techniques for microbial fuel cells to generate electricity from organic matter, and then also it contributes towards the generation of bioethanol, biodiesel, bioethanol, biobutanol, and methane, which is biogas, and as well as advanced materials, production of biocomposites and of late nanomaterials. Industrial microbial biotechnology also contribute towards manufacturing food and feed ingredients like creation of probiotics, single cell protein and food additives, and it also results in production of biopesticides and biofertilizers, which are natural pest control agents, and as well as it produces soil enhancers. It also contributes towards carbon capture and utilization, use of microbes for carbon dioxide sequestration and conversion into biomass or energy. And finally, it is also contributing towards bioremediation, whereby microbial degradation of environmental pollutants and wastewater treatment is undertaken. So, it is a very, very large domain, and we will be discussing many of these applications under various modules and various lectures. So, let us look into some of the industrial microorganisms. Here you can see the picture of penicillium, which is one of the most important industrial microorganism, and this is essential for manufacture of antibiotics. So, let us look into microorganisms in industry. Bacterial metabolism is broadly categorized by the energy source, carbon source and electron donors used for growth. Pathogenic bacteria exhibit diverse metabolic pathways. Metabolites, which are the products and intermediates of metabolism, usually consist of small molecules serving different functions. These metabolites are divided into primary and secondary metabolite categories. In industrial microbiology, these metabolites play crucial roles in producing essential chemicals and biochemicals required for organic synthesis. So, here you can see Clostridium acetobutylicum under 1000x magnification. These species is used widely for fermentation of starch to produce ethanol, acetone and butanol. A later gene from the species was used to genetically engineer Escherichia coli to produce acetone. What are the products from microorganisms? Molecular products derived from microorganisms encompass a broad spectrum of substances suitable for applications in pharmaceuticals, food and beverage production, agriculture and various other industries. This can range from antibiotics and enzymes to various organic acids, alcohols and bioactive compounds, as well as many essential biomolecules like vitamins, amino acids and polysaccharides. So, some of these include antibiotics, enzymes, biofuels, organic acids, vitamins, bioplastics and bioactive compounds. We will have a discussion on these various products and compounds in this lecture. What are primary metabolites? What role they play? So, primary metabolites play a pivotal role in the growth, development and reproduction of organisms. Typically, they serve as central components in maintaining normal physiological processes. They are essential for proper growth and are generating during the growth phase. In industrial microbiology, alcohol is one of the most prevalent primary metabolites utilized for large scale production of alcoholic beverages like beer and wine. Amino acids, notably L-glutamate and L-lysine, are isolated by a mass production of a specific bacterial species, Corynebacterium glutamicum. Another significant primary metabolite in industrial microbiology is citric acid produced by Aspergillus niger. So, here in these photographs you can see various stages of alcohol production in industries. In top figure you can see beer being bottled, and then in the below figure you can see a fermentation vat in a distillery in USA. The secondary metabolites arise typically through the modification of primary metabolite. Unlike primary metabolites that contribute to growth, development and reproduction, secondary metabolites typically emerge toward the end or near the stationary phase of growth. Many identified secondary metabolites serve ecological functions including defense mechanisms such as antibiotic properties and pigment production. Some of the notable examples include antibiotics, bacteriocins and alkaloids. So, here in this figure A, we can see colonies of Actinomycetes bacteria. Both, and then in B we can see Penicillium notatum, both of which are used widely in industries for antibiotic production. C is the Figa paclitaxel, which is an alkaloid used in chemotherapy, is found in several epiphytic fungi. So, some of the important industrial products obtained from microorganisms or with help of microorganisms are traditional products which has been utilized by mankind for over thousands of years, like bread, beer, wine and spirits, and then cheese and other dairy products. Then we have ripening of blue and camembert type cheeses, fermented meats and vegetables, mushrooms, soy sauce, tofu and vinegar. And various bacteria used, for example, we use lactic acid bacteria for producing dairy products. Then we use Penicillium species for the ripening of blue and camembert type cheeses. And bread, wine and spirit, we use fungi, Saccharomyces cerevisiae. Then there are certain fungi, agaricus, then lentinula, which gives us the mushrooms. And then for soy sauce we are using Aspergillus. Then for tofu we use Mucus species, and vinegar we use bacteria, Acetobacter species for production. And then for agricultural applications we have gibberellins, then fungicides, and then insecticides and silage. Gibberellin is produced by the fungi Fusarium mondelliforme, and then fungicides are also produced by various microbes. For example, we can have Coniotherium, and then insecticides like organisms like Bacillus thuringiensis, which produces Bt toxin, and the production of silage, which assisted or mediated by lactic acid bacteria. Then there are certain amino acid like L-glutamine, L-lysine, L-tryptophan produced by various bacteria like Corynebacterium, Babybacterium, Klebsiella respectively. Then we have the various kind of products like enzymes like cellulases, lipases, pectinase, proteases, and then a broad class like carbohydrates like amylase, alpha, beta amylase, invertase, glucose isomerase, lactase, etc. And these are produced by various bacteria and fungi, as can be seen in this table. And then also we have nucleotides which are produced by Bacillus and Brevibacterium species. Then certain other industrial products like fuels and chemical feedstocks like acetone, butanol, ethanol, glycerol, methane are produced by Clostridium, Zymomonas, methazonic species, or even fungi like Saccharomyces cerevisiae. And then we have organic acid producers like Acetobacter, which produces acetic acid, then Aspergillus niger, which produces citric acid, then Rhizopus, which produces fumaric acid, and then gluconic, which is produced by Acetobacter suboxidans, then itaconic acid by Aspergillus itaconicus, and then kojic acid by Aspergillus flavus, and then lactic acid produces by Lactobacillus delbrueckii. So, what are the ideal characteristics of an industrial microorganisms? The ideal characteristics of an industrial microorganism, regardless of its origin, are as follows. It should have genetic stability, ensure consistency in traits over time. Efficient production of the target product. So, we saw so many different kinds of products are produced by either bacteria or fungi, as discussed in the various tables just now. The production of those products should be highly efficient, and the biosynthetic pathway by which these products are made should be well understood. Minimal reliance on external vitamins or growth factors is one of the important features. They should have capability to utilize a wide array of inexpensive carbon sources, so that the cost of production is lowered. They should have the ability to undergo genetic modifications for enhanced traits. Then safety, non-pathogenicity, and absence of toxin production unless intentional for the target product is one important feature. But supposingly our product itself is a toxin, then we will desire that the toxin production is highly efficient in that case. One important thing is the ease of harvesting from fermentation processes, easily breakable cell structure, because most of the time the product may be inside the microbial cell. If the target is or the product is intracellular, limited byproduct generation to simplify subsequent purification process. And then if you have more amount of the product compared to the byproducts, the yield efficiency will also increase. Additionally, advantageous traits like being thermophilic or halophilic will be beneficial in certain fermentation settings. Let us now discuss about pure culture, because nowadays all industrial processes are carried out with pure culture or monoculture. So, we begin with collecting samples. After collecting the sample, the key challenge is determining appropriate growth media and conditions to isolate the target microorganisms. The process begins by suppressing common organisms and promoting rarer ones, often through enrichment cultures in batch or continuous systems to encourage organisms with desired traits. This approach works best when the target trait provides a competitive advantage and acts like a selection marker. Pure cultures are then isolated on selective solid media under tailored conditions. Once isolated, each culture is screened for desired properties such as enzyme production or inhibitory compounds, with strain development expected to improve performance later. Selected isolates are also evaluated for stability and other features like non-toxicity. So, let us discuss the traditional approach for strain improvement. So, there are three methods of natural genetic recombination in bacteria. Number A shows the method conjugation, B shows transduction, and C shows transformation. So, strain improvement plays a crucial role in the development of fermentation industries, enabling cost reduction through increased productivity or lowered manufacturing expenses. Typically, natural genetic recombination methods merge genetic elements from different genomes, forming novel genotypes. Alternatively, mutagenesis is employed and the resulting mutants are screened to identify strains better suited to industrial fermentation. However, these strains might struggle to survive in natural environments and might require specific media for maintenance. Although bacteria lack any form of sexual reproduction, they are able to exchange some genetic material via the process of conjugation, transduction and transformation, as shown in these figures. Conjugation involves direct contact between a donor and recipient via a filamentous protein, a sex pilus. Pulling them together, the donor transfers all or part of its plasmid through the pilus to the recipient, as can be seen over here. Transduction involves a bacteriophage vector delivering genes between bacteria. When the phage attaches to a bacterial cell, its DNA gets incorporated into the host chromosome during its replication. The phage may acquire bits of host DNA and carry them to new hosts. Bacteriophages can also acquire transposons, DNA pieces capable of relocating from one DNA to another and transfer them to the new bacterial cells. So, here we have these phage which is infecting the donor cell, and thereby genetic material is getting introduced from the donor cell into the phage infecting the new host. Then transformation involves the uptake of naked DNA from the environment. Here the phage is not there, neither there is a sex pilus. From the environment into the cell where it gets integrated, this process occurs randomly in natural settings as the DNA fragments originate from live cells. However, only competent cells, which are cells which are ready to accept DNA in a specific physiological state, permit DNA uptake and can incorporate these fragments. So, here you have a donor cell which is releasing DNA, and this is the recipient cell which is competent and receives the genetic material from the donor cell. So, another method is mutagenesis, which involves changes in DNA such as deletions, insertions, duplications, inversions, DNA relocations or variations in gene or chromosome copy numbers. Repeated mutagenesis combined with careful selection and screening has significantly improved many industrial microorganisms. While natural mutation rates in bacterial genes are low, around 10 to the power minus 10 mutations per generation per gene, mutagenesis with agents like UV, gamma and X-rays, then chemicals like ethyl methyl sulfonate, anti-G, nitrous acid or acridine mustards can dramatically increase mutation rates. These mutagens induce base pair substitutions, frame shift mutations or extensive deletions, often disrupting DNA repair. Mutagenesis is commonly used to eliminate undesirable traits or enhance product yield, as seen in the removal of the yellow pigment chrysogenin from early Penicillium chrysogenum derived penicillin. Now, another approach for strain improvement is using rDNA technology or recombinant DNA technology. Here, the cell fusion techniques such as hybridoma formation for monoclonal antibody production have profoundly influenced industrial microbiology. Unlike natural recombination, modern recombinant DNA technology provides precise control methods to create new gene combinations using genetic materials from nearly any organism, living or extinct. This allows specific gene sequences to be transferred between organisms, integrating new techniques into strain enhancement protocols to improve product yields by eliminating metabolic bottlenecks and modifying or amplifying metabolic steps. Microbes can be engineered to produce and secrete a wider range of enzymes, facilitating the synthesis of novel compounds and the use of more cost effective substrates. With no constraints on gene origins, microorganisms now produce proteins from plants and animals, including valuable products like human growth hormone, insulin and interferons. Let us now discuss about the microbial cell factories. Here we will discuss about the characteristics of the microbial cell factories, then approaches to microbial cell factories. So, here in this picture you can see the manufacturing of Cs. Whey from Cs factories are often used in the fermentation media. Briefly, the microbial cell factory approach to bioengineering treats microbial cells as production units by rewiring cellular metabolism to enhance native metabolite production or enable the synthesis of new products. Microbial cell factories are a subset of cell factories which include engineered microbes and plant cells. Initially conceptualized in the 1980s and 1990s, these MCFs aim to enhance cellular productivity and metabolite yields through strain engineering. By employing targeted strain design, MCFs can produce both native and non-native metabolites. Moreover, they streamline synthesis cycles and simplify product separation processes. So, traditional strategies like eliminating shunt enzymes, modifying pathways and balancing reducing power and ATP often face challenges such as toxic byproducts, low pH and extreme temperatures which limit productivity. MCF technologies address these issues by integrating traditional methods with synthetic biology, systems biology and evolutionary engineering. The approach involves selecting target products, analyzing market demand, identifying cost effective carbon substrates and optimizing host strains and pathways to enhance yield. Let us look into the various approaches to designing biosynthetic pathways in microbial cell factories. So, here you can see the existing pathway within the host. We call them the native existing pathways. So, then we have these pathway created de novo. So, this is the de novo blue colored pathway which is non-native created pathway. And then we have pathway from say species A. So, we clone the genes from that species and put it into here. Then we may have another pathway from species B and another pathway from species C. So, basically we may have three approaches. One is utilizing the native existing pathway, or we may create a non-native pathway de novo, entirely new, or we may borrow from various organisms to build up a network of pathway like taking from species A, B, C as in the center. And then ultimate result is we have a substrate which enters the cell, and these substrate is acted upon by any of these pathways, the native, non-native, de novo, or the non-native transferred existing pathway, and then transform these into products which are of having economic value. So, these cell considered as a factory on its own, because you put in raw material and then you get product, and that raw material is converted into products by numerous pathways as discussed over here. Now, in silico modeling of E. coli reveal its ability to produce 1777 non-native compounds via heterologous enzyme introduction. If we introduce heterologous enzymes into the E. coli system, we can produce 1777 non-native compounds. Then there are 279 of which have notable commercial value. So, imagine one single cell having the, by introduction of heterologous enzyme systems, suddenly gains the capability of producing roughly around 2000 different chemicals, and then out of which roughly around 300 have high commercial value. So, that is the potential of microbial cell factories. So, what are the characteristics of microbial cell factories? After the selection of a valuable compound and substrate, a cell factory is selected, that is basically the host microorganism. For example, in this case E. coli can be a cell factory for these 279 products having commercial value. The criteria for selecting the starting strain comprise the following steps. Number one, the strain should be easier manipulation of the respective organism. It should be easy to be manipulated. Whether the selected product is native, partially native or non-native, we should have that knowledge. Suitability and sustainability for large scale production, because we are speaking about industrial scale production. So, the strain should be suitable for that large scale production. It should show optimum growth and desired products formation on simple culture medium. The culture that we are going to use should not be very costly. It should be simple and abundant and low cost. It should have metabolic capacity towards the end product. Then bioprocess compatibility is very, very important. Then the recovery should be cost effective, and then purification procedures to the maximum extent economical and simple. Model organisms like E. coli and Saccharomyces cerevisiae are widely used due to advancements in genome sequencing, because we know the entire genomes of these organisms, and then we can understand whether that pathway is native or non-native. So, we can do metabolic modeling, and genetic tools like CRISPR-Cas9, which have simplified and reduced the cost of the strain engineering, are very well established in both these two systems. Let us now discuss about the various approaches to microbial cell factories. So, here you can see that different pathways which we have just discussed about the native existing pathway and the non-native existing pathways, and non-native created pathways. So, how we are going to take decisions at various levels. So, first we need to consider the product, targeted product in our mind, and then for producing that particular product we need to select the host strain, and once we have this strain in our hand, we will try to examine whether this particular strain is a native producer of this particular product? If the answer is yes, we can go for the production. If the answer is no, we will try to find out whether that pathway which produces this particular product is known in nature. Then we may go for two approaches. One is reconstruct the pathway in the host, and then this will help us in the formation of the product. Or if it is not known, we go for metabolic pathway discovery, and then we try to find out the pathway and reconstruct the pathway in the host. And in this, we will go for the retrobiosynthesis and then construct the pathway in the host, and finally the product will be formed. So, these are the three various approaches by which we go for microbial cell factory and strain improvement production. So, some microorganisms naturally produce significant amounts of valuable chemicals without requiring initial metabolic engineering. So, here they have the native existing pathway. So, here you can see glutamicum under 10,000 magnification visualized using scaling electron microscopy. This organism has been widely used to synthesize glutamate and lysine. So, this is that C. Glutamicum species which produces glutamate and lysine. Similarly, there are other species like Bacillus and Lactobacillus which produce L-lactate. Klebsiella pneumoniae synthesizes 1,3-propan diol from glycerol. Then we have Manhemia produces succinic acid, and Actinomycetes generate antibiotics and polyketides. These are all native existing pathways within these microorganisms, and we can directly use them for the production of these particular targeted products. So, we only have to go for optimal host selection and engineering further to improve the production efficiency if the production efficiency is less. In the second approach, the non-native existing pathway. So, some target chemicals cannot be synthesized by microbial host due to missing biosynthetic genes, because those genes are missing over there. So, we can resolve this by incorporating enzymes and pathways from other organisms or metagenomes. So, we have databases like KEGG, Metapsych and Brenda, which provide detailed information on metabolic pathways, enzymes and genes, enabling pathway reconstruction by integrating external genes. For products with incomplete pathway data or poorly functioning enzymes, novel enzymes can be identified through genome and transcriptome analysis of natural biosynthetic gene clusters. Then we have the non-native created pathway. Metabolic engineering has enabled the production of many chemicals using existing biosynthetic pathways. However, to fully replace petrochemical refineries, microbial cell factories must also produce non-natural chemicals or natural products with uncharacterized pathways. Retrosynthesis is a method in organic chemistry that works backward from a target chemical to its precursors, which can now be adapted with computer aided tools to design biosynthetic pathways. These pathways can be constructed de novo by identifying suitable enzymes from heterologous sources. For example, a de novo pathway was developed for producing beta-lactam, valorolactam and caprolactam using omega amino acids as precursors in these pathways. C. propionicum derived B-alanine coenzyme A transferase activated the omega amino acids, leading to their spontaneous cyclization into lactams. So, let us now have a overview of the applications of MCFs. We have various products over here like acetone, citric acid, succinic acid and so on, and then we have the organisms which can produce them. For example, acetone is produced by Clostridium acetobutylicum. Then we have citric acid, which is produced by Aspergillus niger. E. coli produces succinic acid, then 1,3-PDO, 1,4-BDO. Then isoprene produces by Saccharomyces cerevisiae, and again we have isobutene produced by E. coli. Now, many of these have been already commercialized. Some are under various stages like preparation, demonstration, and also some of them have been again commercialized. Now, we use various kind of feedstocks, and in one of the slides we discussed that the substrates feedstock should be very low cost. For example, we are using corn or corn sugars or sugar, cellulose or glucose and sucrose, and various companies which have already taken lead in production of these particular chemicals. And then we have more like production of ethanol from sugarcane using Saccharomyces cerevisiae. Then we have farnesene, and then we have butanol, isobutanol. These are all already commercialized. Let us now move to section 4, where we will be discussing the fermentation process overall, having the various upstream processes and the downstream processing. So, here in this picture you can see an industrial bioreactor, filtration and sterilization equipment. Fermentation in general refers to the chemical transformation of organic substances into simple compounds by the action of enzymes produced by microorganisms such as yeast, molds or bacteria. It can be classified on the basis of substrate used, like solid state fermentations, submerged fermentations. While solid state fermentation are practiced, most fermentations employed liquid media, often termed as broth under aerobic or anaerobic conditions. Most fermentation processes involve stirring, aeration and adhere to aseptic protocols, exception being most alcoholic fermentation. Let us discuss about the solid state fermentation first. So, here is a diagrammatic representation of an solid state fermentation process. So, here we have a fermenter with an insulating jacket, then this is stacked with certain trays having the substrate, and then there is a air supply from below and there is a air outlet, because the air supply will be continuous and the air has to be let out from the system. So, it uses natural raw materials as carbon sources. So, these are the natural raw materials placed in these trays, which may be cassava, barley, sugarcane bagasse, oil cakes, palm kernel, soybean groundnut, fruit pulps, seeds, tamarind, sawdust, coffee husks and tea waste. So, you can see from these that many of these are in general agro wastes, for example, or it may be a waste from the timber industry. It is widely used for producing biomolecules such as metabolites in the food, pharmaceutical, cosmetic, fuel and textile industries. Now, in the submerged fermentation, the fermenter is different. So, here you can see a fermenter which has a water inlet. So, basically this is a water jacket which keeps the temperature of the fermenter fixed. So, you have these temperature control valve over here. Water is heated to a desired temperature, which will carry the heat and provide the warmth to this fermenter. There is an optimum temperature condition at which these microbes will do the fermentation. And of course, there is continuous supply of air from below, and then you have certain probes over here, the temperature probe, which will keep track of the temperature of these media, and then you have a stirrer which will keep on mixing the media. So, submerged fermentation techniques cultivate microorganisms in liquid broth. Selected microorganisms are grown in closed vessels containing a broth rich in nutrients and high concentration of oxygen. Substrates are utilized quite rapidly and hence is constantly replaced, or the media is constantly supplemented with nutrients. Bacteria that require high moisture content or high water activity are best suited for submerged fermentation. submerged fermentation is primarily used in the extraction of secondary metabolites, they need to be used in liquid form. So, now let us discuss about the fermentation media a little bit more detail. Most fermentation processes primarily utilize liquid media, which is often termed as broth, although there are instances of solid substrate fermentations as we have discussed just now. So, here in the left we can see algal fermentation media in lab scale, and then in the other figure we can see the molasses, which is one of the most widely used carbon source in the industrial setting. The fermentation medium is crucial, needing to meet both the nutritional needs of the microorganism and fulfill the technical goals of the process. Thus, the nutrient composition should be carefully devised to stimulate and the production of the desired output, be it cell biomass or a specific metabolite. In almost all fermentations except those involving solid substrate, substantial volumes of water are used in formulating the media. Apart from these water, let us see the composition of the fermentation media. So, essential components of a typical medium include a carbon source, which serves as both an energy source and carbon unit for biosynthesis, along with nitrogen, phosphorus and sulfur sources. Additionally, minor and trace elements must be provided, and some microorganisms may require supplementation with specific vitamins like biotin and riboflavin. Aerobic fermentations rely on a constant supply of molecular oxygen, while even some anaerobic fermentations necessitate initial aeration of the media. Media typically includes buffers or employ pH control through the addition of acids and alkalis, and antifoam agents are very essential. Certain processes may require the introduction of precursors, inducers or inhibitor compounds at specific fermentation stages. So, what are these precursors and antifoams? In certain fermentations, specific precursors are added in controlled amounts and in relatively pure forms. So, some of the examples are phenylacetic acid or phenylacetamide in penicillin production, D-threonine in L-isoleucine production by Serratia muscarinens. Then we have anthralinic acid in L-tryptophan production by Hansenula and Omela. Then antifoams are also essential. There is foam formation when media proteins denature at the air broth interface, leading to issues like filter blockage, contamination and microorganism release. Antifoams being surface active agents reduce the surface tension that binds foam, thereby improving throughput. Natural antifoams encompasses plant oils like soya, sunflower and rapeseed oils, deodorized fish oil, mineral oils and tallow. Synthetic antifoams primarily consist of silicon oils, polyalcohols and alkylated glycols. Then there are inducers and inhibitors, which are very essential in fermentations involving genetically modified microorganisms. Inducers are frequently essential for the expression of clone genes in these genetically modified organisms. When turned on, can impede their growth due to exceptionally high levels of transcription and translation. Consequently, these clone genes incorporate inducible systems that initially maximize growth to establish high biomass density. Subsequently, the clone gene can be activated by introducing a specific chemical inducer. Inhibitors are employed to redirect metabolism towards the desired product and curtail the formation of other metabolic intermediates. Inhibitors halt a pathway at a particular stage, preventing further metabolism of the target product. For instance, in the production of glycerol by Saccharomyces cerevisiae, sodium bisulfite is used as an inhibitor to redirect metabolism. One of the important point is inoculum. In industrial fermentation, the term inoculum refers to the initial culture of microorganisms, whether bacteria, yeast or fungi, that is introduced into a fermentation process. So, in this picture we can see the preparation of a wine yeast starter culture with yeast getting rehydrated. These initial culture serves as the starting point for the production of desired products through fermentation. The inoculum contains a high concentration of the selected microorganisms and is used to inoculate a larger fermentation vessel, providing the microorganisms with the necessary conditions to grow, multiply and carry out the fermentation process. on a larger scale. The goal is to achieve optimal conditions for the production of various substances including biofuels, enzymes, organic acids or pharmaceuticals depending on the specific industrial application. Let us now discuss about one of the important apparatus or equipment in this fermentation process, the fermenter. The primary role of a fermenter is to create an optimal environment where an organism can efficiently generate a desired product, whether it is cell biomass, a metabolite or a bioconversion product. Most fermenters aim to maintain high concentrations of biomass, crucial for many fermentation processes. Control strategies vary based on the specific processes and its objectives. The performance of a fermenter hinges or relies on various factors, with key control parameters being agitation rate, oxygen transfer, pH, temperature and the foam production. So, in this picture left you can see the fermenting and conditioning tanks over here, and then on the other picture we can see the lab scale fermentation tank with mammalian cells, and this is just to give an idea that fermentation may also be carried out by non-microbial cells like the mammalian cells in this case. There are two types of fermenters. One is the smaller ones which are used in the lab, and the bigger ones like these which are used in the industrial settings. So, laboratory scale fermentations might use simple bottles or conical flasks that can be agitated to provide aeration when needed. These vessels are typically sealed with cotton wool or a styrofoam bung to prevent microbial contamination from outside, but this sealing method can lead to evaporation losses and limited gas exchange. Consequently, specifically designed vessels for fermentations are generally preferred even at the laboratory level. In industrial application, fermenters with capacities reaching several 100,000 liters are utilized. These are typically purpose built and tailored for a particular process, although some flexibility might be required in certain cases. Their design, construction quality, mode of operation, level of sophistication are largely dictated by factors like the production organism, optimal operating conditions for the target product, product value and production scale. The primary considerations include reliability and the imperative to minimize both initial investment and ongoing operational expenses. Let us now discuss about the control of physical and chemical conditions. Controlling the key parameters of a bioreactor such as temperature, pH, pure oxygen and pressure are essential to maintain cells in a physical and chemical environment, optimizing their performances. The majority of cells have an optimal temperature of operation. Temperatures higher than these optimum can quickly have a dramatic effect on the cell viability, while lower temperatures can result in a slower cell metabolism. This optimum is maintained by a temperature sensor, a water jacket on the bioreactor and a temperature control unit. Similarly, cells have optimal operational pH maintained by buffers like bicarbonate buffer. Most bioreactors or fermenters employ a proper agitation system which allows the media, the cells and the gases to be maintained in an ideal mixing state. Additionally, aerobic fermentations may require optimal concentrations of molecular oxygen, which is maintained by measuring the oxygen transfer rate, representing the rate at which oxygen can be delivered to the biological system. In lab scale fermentation, after selecting a microorganism for a specific process, initial optimization is conducted in laboratory scale conditions, typically using fermenters ranging from 1 to 10 liters. Here you can see a lab scale bio fermenter. This involves analyzing media composition, exploring various feeding strategies, whether we go for batch, fed batch or continuous fermentation, and determining the most suitable fermentation system types such as tire tank, airlift, packed bed, solid state, hollow fiber, etc. Other critical considerations encompass reactor setup and the management of pH, dissolved oxygen, foam and temperature control. Once optimal product yield is achieved in the laboratory, The process undergoes scaling up initially to pilot scale, ranging from 10 to 100 liters. So, from 1 to 10 liters, we go to 100 liters, and eventually to industrial scale, which will go up to 1000 to 100,000 liters or more, depending on the specific process capacity of the plant. So, scaling up is very, very important. So, here you can see a small flask around 500 ml, then we are going for 5 liters, then 50 liters, and then 500 liters. So, as we move from a small volume to a large volume, this process is called as scaling up. Fermentation scale up refers to the process of increasing the volume of a fermentation process from laboratory or pilot scale to a larger industrial scale production. It involves replicating the fermentation process in larger vessels while maintaining or improving productivity, yield, and product quality. So, the yield and quality in these small and the yield and quality in these large one should ideally be similar, if not same. Large scale fermentations lower production costs through economies of scale, enabling efficient production to meet market demand. Adapting processes to maintaining product quality and consistency during scale up is essential. Several key factors influence yield during the scale up process. Number 1, differences in inoculum propagation procedures in the quality and quantity of inoculum used. Selection of the medium. Cost limitations at industrial scales might involve cheaper nutrient sources. Industrial scale sterilization methods might cause greater degradation of heat sensitive compounds, affecting the medium quality. Development of gradients in larger fermenters, including nutrient, temperature, pH, and oxygen, which were absent in smaller well mixed systems. Alterations in foam generation, shear forces, and carbon dioxide removal rates. Variations in these factors can significantly affect operational conditions and subsequently influence product yield compared to laboratory scale fermentations. Let us now have a discussion on the downstream processing, where we will be discussing about cell harvesting, disruption, and the product recovery. So, here we see a industrial centrifuge used for multiple purposes in downstream processing, and you can see the size of it, which is very, very big compared to the laboratory centrifuge. So, downstream processing encompasses all post fermentation procedures after we take out the product out of the fermenter, aimed at recovering the target product efficiently, consistently, and safely while maximizing yield and minimizing cost. The process depends on fermentation protocols and microorganisms properties such as morphology, size, and cell wall rigidity, which influence filterability, sedimentation, and homogenization. Fermentation byproducts, media impurities, and additives like antifoams can hinder downstream processing and product analysis. Effective purification strategies must account for upstream and downstream factors, including product properties, concentration, and location, as well as stability to prevent degradation. So, let us start with cell harvesting. The initial stage in downstream processing for suspended cultures involves solid liquid separation to extract cell from the spent medium. Each fraction can then undergo further processing determined by whether the product is located intracellularly or has been secreted into the periplasmic space or the medium. The selection of solid liquid separation method is influenced by factors such as the microorganism size and morphology, single cells, aggregates, or mycelia, as well as the specific gravity, viscosity, and rheology of the spent fermentation medium. Some of the various methods to achieve this are sedimentation, which lets the cells settle at the bottom of fermenters with passage of time; broth conditioning, which uses flocculants to make the cells clump together and later sediment; centrifugation, which employs centrifugal field; then filtration, which employs porous material like cloth, glass wool, or cellulose to retain the solids. The next important downstream operation is cell disruption. Disrupting the microorganisms to release intracellular products like enzymes and recombinant proteins can be a challenge due to the need to breach the cell wall and membrane. Mechanical methods that can be applied for cell disruptions are liquid shear, which exposes cells to sudden pressure drops by discharging them through a narrow valve. Manual grinding, which causes cell breakage due to shear forces, grinding and collisions. Ultrasonic disruptions, which uses cavitation. Alternative chemical techniques are also employed, like autolysis, which uses the cell's own enzymes to break down the cell components. Osmotic shock, by treating cells with 20% sucrose, then rapidly suspending them in cold water. Organic solvents like acetone, butanol, chloroform or methanol, or detergents like sodium lauryl sulfate or Triton X100, which disrupt the membrane dynamics. Then utilization of cell wall degrading enzymes like lysozyme, which target bacterial peptidoglycan. Then using antibiotics like penicillin and cycloserine. And then the next is important operations is the product recovery. So, extracellular products are recovered from the clarified medium, whereas disrupted cell preparations are utilized for both intracellular products and those residing within the periplasmic space. In case of high level expression of certain recombinant proteins, inclusion bodies formed are released through cell breakage. So, here in this figure we can see the principle of salting in and salting out techniques based on increasing salt concentration. So, here the salt concentration is increasing. So, the salting in sets in the beginning. So, then the a phase is reached where there is say maximum solubility, and then the solubility goes down after this salt concentration. So, this is the salting out phase, where you can see that these salt molecules which were binding helping in the protein interactions, particularly the hydrophobic regions. So, as the salt concentration increases, and the salt's concentration will induce the hydrophobic regions of the proteins to come together, and then finally the coagulation will start happening, and they will become very heavy, and then they will precipitate. So, this is the salting out technique, which is shown here diagrammatically. Post cell disruption centrifugation typically separates soluble products from the cell debris. Conversely, products which remain in the supernatant can be recovered using salting out, which involves precipitation using inorganic salts, ammonium sulfate in solid or saturated solutions. Organic solvents like acetone, ethanol and isopropanol can also reduce product solubility at low temperatures, causing their precipitation. Then there are chromatographic techniques which are used for high value products, rely on factors like molecular weight, isoelectric point, hydrophobicity and biological affinity. Here in this picture you can see operators preparing a programmable chromatography skid with packed column. These methods can be scaled industrially to ensure capacity, recovery and resolving power, especially for final purification. Protein integrity is protected by running columns at 4 degrees centigrade and managing pH changes, dilution and chemical additives. Membrane separation methods such as dialysis and electrodialysis removes low molecular weight solutes and ions using size selective membranes or ion exchange groups with electrodialysis also applied in water desalination. Then another process is the crystallization. Product crystallization encompasses evaporation, low temperature treatments or introducing a reactive chemical to the solute. Solubility reduction of the product can be achieved through various means like adding solvents, salts, polymers such as non-ionic polyethylene glycol, then polyelectrolytes or adjusting the pH. Drying entails applying heat to the wet material and eliminating moisture as water vapor. This process typically aims to preserve the biological activity of the product. Factors influencing drying include the physical characteristics of the solid liquid system, inherent properties of the solute, environmental conditions during drying and heat transfer mechanisms involving direct contact, convection or radiation. So, with this we come to end of this lecture. Thank you for your present attention.